Hi guys, so I'm extremely excited to share this new platform with you and see how it works for us as a class. What's great about this is that you can pause and you can rewatch this video as many times as you'd like, and then if you don't understand anything you can just go back and rewatch it again. I will be sharing quite a few resources with you on our group, and then we'll also be doing some old exam questions as we've been doing in class, just to make sure that you guys understand the topic and that you understand the type of questions that they can ask in an exam paper. So the topic that we'll be starting with today is genetics and inheritance. Now, as in your exam guidelines, it is in paper 2 and it's about 45 marks, which is quite a lot. That's about a third, just under a third of paper 2's marks. So you guys do need to make sure that you understand this work, and it can get a bit complicated as soon as we get to the genetic crosses, but if you remain focused and have an open mind, you won't struggle with it. It's very logical. That is why I like genetics. It's logic. It's a lot of logic, and it's straightforward. So if you know the basics of it, you'll be fine. We're looking at it at a very basic level now with the matric syllabus, but these basics you will carry on if you go study further in university and you do genetics. If you understand this, you'll be fine then. So what is genetics? It is the study of heredity and variation in living organisms. So basically what that means, it is the inheritance of characteristics that you'll get from your parents because of their genes that they have. Now, who started the study of heredity and variation? So a lot of what we know today can be attributed to this guy over here, and his name is Gregor Mendel. Now, he was an Austrian monk, and he did a lot of experiments on pea plants. Now, a lot of his work that he did is used today, the basis of it, but sadly none of his work was ever used while he was still alive. It was only after he died that people made use of his findings. So why use pea plants? So firstly, Gregor decided on pea plants because they grow quickly. So whatever he was looking for, he could see the results very quickly after he's tested the plants, because they grow quickly, obviously. Then they have easily identifiable features or characteristics. Okay, so that means that when you look at pea plants, they'll have either or. So in this case, they'll either have smooth, round seeds, or they'll have wrinkled raisin-like seeds. The seeds will be yellow, or they can be green. The flowers can be purple, or they're white. And then you also could look at the seed pods themselves, position of the flowers, and then whether it was a tall plant or it was a short plant. These are all of the features, the easily identifiable characteristics that Mendel looked at. Then another one is that they could either self-pollinate, or what Mendel used was the fact that they could cross-pollinate. So this was very helpful to him because he could literally cut off the anther of a purple flower and take the pollen and deposit it on a white flower and then cross-pollinate them like that to see what features would come out. Now, with his pea plant experiments, Mendel was able to formulate two laws. So the law of segregation and the law of independent assortment, but we'll not be looking at those yet. I just want to run through these definitions, these terms that we'll be using throughout this chapter. They are quite important to know and understand the work. So the first one we'll look at is allele. So what is an allele? It is an alternative form of the same gene. They contain different info about the same characteristic or trait. So usually you will get one from your father and one from your mother. So let's look at this diagram. And so this is a chromosome in one cell, and this is the other. You could also just imagine that there are two more because this could be at the end of meiosis 2, specifically telophase 2. So we just said that an allele is the same gene, but a different characteristic or trait of that gene. So on this chromosome, this light blue area would be the allele. So if we use this diagram as an example, this area over here would be the allele, the gene would be for eye color, and then the trait that this specific chromosome is carrying on that allele is for brown eyes. So this one could be, for example, for brown eyes, and this one for blue eyes, because it is the same gene, just with a different trait or characteristic. I hope you guys understand that. Now if we go back, and a locus, so what is a locus? It is the area where one gene is located. Now going back to this diagram again, the gene for eye color, specifically on this chromosome, sits right here. That is the location. So locus could mean location, the location of the gene. Okay, so the location of this gene is on this chromosome right here, and it is the gene for eye color. The gene for hair color on this chromosome, the locus sits over there. Now, homozygous and heterozygous walk hand in hand with dominant, recessive, and all of these traits. So if I go through these definitions and you guys do not understand them yet, do not worry, you will by the end of this video, hopefully. So what does homozygous mean? So we know by now that homo means same, and then zygous is for zygote. So you know a zygote is formed after fertilization has occurred, you get a zygote. So the same zygote basically. So when two alleles are the same for one trait, example, a red flower, the color red from both parents. So both parents carry the trait for a red flower. That's a horrible flower. So they are the same color, the same trait. Then heterozygous, hetero means different, and then zygous, you know what that means. So when two alleles of a pair of genes are different for a trait, example, one blue flower parent crosses with a red flower parent. So you've got a parent that has the trait for a red flower, and then you've also got a parent that has a trait for a blue flower. So in this case, they are heterozygous because they are different. Then we also look at dominant trait. So it is the stronger trait, and it will always overshadow the recessive trait. So think of something that is dominant, it is overpowering, it will overshadow, it takes over. That is what a dominant trait does. Then the recessive trait is the weaker trait, and it will only come through if there's two of them. I'll explain this to you now. And then the recessive trait is always dominated by a dominant trait. So if we use Mendels pea plants as an example here, so a tall plant versus a short plant. So a tall plant is always dominant over the short plant. And how we show this in genetic crosses is by using the first letter of the dominant trait to show this symbol. So in this case, tall is the dominant trait, so the first letter is a T, so it is shown as a capital letter T. The short plant will then be represented by a lowercase letter of the dominant trait. So in this case, short will be a lowercase t. Now remember, you get traits from both your parents. So one from the maternal side and one from the paternal side. So your papa and your mama. Your spelling and writing. From your papa and your mama. So let's say your mom has the trait, or in this case, the pea plant, the one from the mother or the female has the trait for being tall. So that is the dominant one. And then the father has the trait for being short. So it will be written like that. So one from your mom and one from the dad. Now remember, I told you the dominant trait always overshadows the recessive trait. So that is why the plant will be tall and not short, because it has that dominant trait. Now in this case, because there are two different ones, we call this heterozygous. Okay, because they are different. If it was, for example, the mom has the tall dominant gene as well as the father, they would be the same, so we would call that homozygous. And then if, let's say, the mom was short and the father was short, then you'd have a homozygous short plant, because there is no dominant trait to overshadow that one. It's just these two. And that is why here in the definition, weaker trait and will only come through if there's two of them. So only if there's two recessive traits will it be homozygous short, and will the plant actually be short. In this case, if it is heterozygous tall like this, the plant will still be tall, because that dominant trait completely overshadows the short one over there. And if it's homozygous tall, obviously there's two dominant traits, so it will obviously be tall. Now, just for interest's sake, you can go look at this on the internet yourself. There's quite a few characteristics or traits that you can look at to see if it's dominant or recessive. But in humans, just for interest's sake, a cleft chin, so having a butt chin basically, is dominant over having no cleft. So if you've got a cleft chin, you have a dominant trait. If you have a widow's peak, so what is that? That is basically a V shape of your hairline on the forehead. That is a dominant gene. If you have a straight hairline, that is a recessive gene. Brown hair or darker hair is dominant over blonde hair. Brown eyes dominant over blue eyes. So just for interest's sake. So for example, if you have a recessive gene, let's use this as an example. If you have a recessive gene, for that recessive gene to show, it has to be homozygous. So let's say having a widow's peak is represented by a capital W, and having no widow's peak is represented by a lowercase one. So in this case, it being recessive, it has to have two lowercases. Whereas if it's dominant, it can be a capital W and a lowercase w, or it can be homozygous dominant with two W's. Same with brown eyes or blue eyes. So if you've got brown eyes, it will be a capital B with a lowercase b, because it can be heterozygous, or it can be homozygous dominant like that. But if it is recessive, it will definitely be homozygous recessive with two lowercase b's, because as soon as a capital B comes in, it's going to overshadow that completely, and you will have brown eyes. The next two definitions that we'll be looking at is the genotype and phenotype. So a genotype is the genetic makeup of an organism and is the information that is present in the genes. So geno stands for genes. Okay, this is a genetic makeup of an organism and is the information that is present in the genes. This is a trait that cannot be seen when looking at an organism. So these traits would be, for example, a tall plant, or a short plant. That would be the genotype of a pea plant. Okay. It being tall or short. Now the phenotype. So pheno means the physical appearance, so the features. Pheno, features. That as PHF, pheno sound, features. Okay, what you see when you look at an organism. So this is determined by the genotype. Obviously, if the genotype looks like this, you will see a tall plant. If you see a genotype like that, it will also be tall. If you see a genotype like that, the plant will definitely be short. Okay. So Mendels Laws, the law of segregation, and the law of independent assortment. Now, what does that mean? Now, the law of segregation means that during meiosis, the chromosomes separate, leaving each cell with a single allele for each trait. When two alleles of a pair are different, one is dominant over the other recessive trait. So this is what Mendel found, basically. Now, if we look at meiosis, at the end of meiosis 1, so this would be meiosis 1 and this would be meiosis 2. So at the end of meiosis, you would have had, at the end of meiosis 1, you would have had homologous chromosomes separating. Okay, so these guys were homologous chromosomes. Then they separated. One went to the one pole and one went to the other pole. Now on each of these chromosomes would be an allele. Don't get confused with crossing over and alleles now. So remember, an allele is different characteristics or traits for the same gene. So on this side of the chromosome, there's a trait for, let's use hair color. Okay, and on this side is the same gene, also for hair color. This side might be for brown hair, and this side might be for blonde hair. And same here, remember these were homologous chromosomes, so the locus for hair color, the allele, would sit on the same spot basically. So this could also be for brown or blonde hair, depending on what you think. Okay, so that is what the law of segregation means, that there will always be an allele for the same gene, just that the trait will be different, either for brown or blonde hair. Then that brings us to the law of independent assortment. So what this means is, at the end, let's say you've got, so once again there's the allele for hair color. Okay, so red represents hair color. Now you can also have another trait that's also on this chromosome, or another gene that sits there for the allele. Now we can say that this represents eye color. Now what the law of independent assortment means is that one allele, so this allele over here, will not influence the allele over there. So whatever hair color is here will not influence the eye color. That is what makes meiosis and genetics so interesting, is that there's such a large amount of variation that can occur, because there's, brown hair or blonde hair can be combined with brown eyes or blue eyes. This combination, it can go either way, if you understand what I'm saying. So the law of independent assortment means that whatever hair color there is, it won't influence the trait for eye color. That is what independent assortment means. And this is where we will stop with the first video. Let me know if you guys have any questions and we'll look at that.